Greetings. Welcome to Scientific Reasoning Practice Test. This is part two. Part one was our content that we did on Tuesday, and now we're going to see what we remember. So I'm super excited because we did start a moment or two late today because we're also now live streaming on Instagram. So how exciting. And ultimately, we're going to get TikTok on there too, but we had some issues with that today. So I am not going to hold anybody up. We're going to dive right in. We're going to hopefully get through this in about, I would say, an hour and 15 minutes would probably be the expectation. Just for your clarity here, if you signed up via Zoom, when we have questions that we go through, if you put your answers in the chat box, only I can see them. If you are streaming on any other service, any of our socials, everyone will be able to see your responses. But I still highly encourage you to put your answers in there. That helps support engagement. It helps you remember the ones you got correct, the ones that you need to work on, things like that. So I strongly encourage that you do participate, whether others can see your answers or not. All right, so diving right in, I always like to kick off with all our QR codes up on the screen so that if you want to access some of the other things we have here at Archer Review, you have easy access to them. So you can see you've got the free live webinars like you're participating in today. You can watch old webinars. You can gain access to our question bank. That's that next QR code. And I'm so happy to share that as of last night, we hit the 2,000 question total. So we're over 2,000 now. It's super exciting. As a matter of fact, I really just want to show you this question bank. This is the one that I get the most feedback on over anything, is how amazing our question bank really is. So if I want to go in here and I want to, let's say I want to create a test on scientific reasoning, I can go on in, let's say I want 45 questions. I find my scientific reasoning topic and I scroll down and I pick the subtopics that are in there so it will craft my test for me. I'm not sure why that one's showing 0. We do have some in there, but maybe a little glitch today. Nonetheless, it creates our questions for us, right? And we can scroll through here. We can reveal answers. This is asking us to arrange things. We can go on to the next. Let's answer one. Let's see what happens if we answer one. So this one's asking us, why is a control group important in an experiment? Well, that's because it's providing a baseline for comparison. The way I set it up was that it would show me the answers right away. So it shows me the correct answer. It gives me our rationale as to why the others are incorrect. Tells me what my learning objective was. It also even tags a video to help support that answer. Which is just super cool, as well as the additional info to just really support the topic in general. And then the other thing, while I'm in here, I might as well show you, which I think is so cool, is our on-demand videos. So the way that we have it set up, you can't actually see the video, but what I want to show you is if I was to go into a video, not only do you have the video, not only do you have the video, but you also have access to handouts and cheat sheets. I love these cheat sheets. These cheat sheets, look at that. Isn't that amazing? They're so pretty. I feel like they're art. So I wanted to point that out. You also have all the slides. So the on-demand video then gives you all the slides that you see in the video. So super cool stuff. I know I got sidetracked from the question bank because I was so excited to show you those items as well. But I just wanted to make sure you guys see them because it's just they're so helpful and they can really help you achieve your goals. So that led us to the next QR code, which obviously was the on-demand videos. And so we looked at those. Also remember that you have tutoring. I'll show you what your tutors look like. And also we have packages. So let's take a look at our packages. We have the SurePass and the SurePass Pro. So the SurePass is that question bank that I just went through and those on-demand videos. And the SurePass Pro adds in an hour of private tutoring. Your tutors are my colleague and friend Tammy. She does English and reading, myself for science, and then Brandon for math. And then as I had kind of skimmed through, but I'll come back to, we are on all the socials. So find us on Facebook, Insta, TikTok, and you can also email us at pre-nursing. Okay, so definitely check that out. Our packages are really affordable. I'm so proud of that. I'm so proud of working for a company that gives you a fantastic product at an affordable price, and all to help you reach your goals. So that gets me excited. All right, so as promised, we have our practice test. And so what I like to do with these is I'll read through the question, I'll read through the answers with you, and then I have a timer that will go for about 40 seconds. And that mimics kind of the pace of TEAS. And then when that timer goes off, we will reveal the correct answer. We'll talk about why that's correct. And we'll also look at the rationale for why the other answers are incorrect. Okay? All right, so I see lots of comments going out there. Thank you. So people are talking about the Q Bank, and yes, it is amazing. And you do get that free trial, so definitely check that out. And oh, somebody's going to do a SurePass combo. Oh, great. If you do the combo, then maybe we get to work together on a tutor session. That would be fantastic. Okay, here we go, guys. Let's get started. So we're kicking off with scientific measurement. That aligns with S4.1. All right, which of the following correctly orders these metric prefixes from smallest to largest? A, deci, micro, nano, milli, kilo. B, nano, micro, milli, deci, kilo. C, nano, milli, kilo, deci, micro. D, milli, nano, kilo, deci, micro. Got 45 seconds. Get those answers out there. All right, let's see what we got. Answer B. And for those of you that put your answers out there, I think everyone got that correct. So remember that if we're going from smallest to largest, we had that silly statement, now many millipedes crawl down mountains. And I literally like envision millipedes crawling down a mountain to help me remember that. So keep that in mind. Answer A was incorrect because milli needs to come before deci. Answer C is incorrect because milli was incorrectly placed after kilo. And then answer D is incorrect because nano is smaller than milli. All right, we're going to move to our next one. A student is measuring the volume of a liquid in a graduated cylinder. To obtain an accurate reading, where should the student measure the liquid's volume? At the highest point of the liquid's surface? At the lowest point of the meniscus? At the middle of the liquid's curve? At the top of the container? Get those answers out there. Okay, our correct answer is B. We want to measure it at the lowest point of the meniscus. Okay, and this I pulled this visual from our content on Tuesday. So you want to see that arched surface or that concave surface, and you want to go right at the lowest point of it. Okay. So answer A was saying at the highest point of the liquid surface. And what happens there is it actually overestimates the volume then. C, I could see where someone would possibly have tried to say C. Because it kind of sounded like it was saying meniscus, but B was certainly a better answer because it was using that language meniscus. And then D was asking at the top of the container. Obviously, no, we wouldn't measure at the top of the container because that's not where the liquid actually was. Yeah, and C, I think the wording for C, I saw a couple comments about that. I think the wording for C just really kind of made you second guess yourself. So I could see why that would be an easy mistake for sure. Okay, a nurse needs to administer 1 gram of medication to a patient. However, the pharmacy only provides the medication in 250 milligram tablets. How many tablets should the nurse administer? A, 2, 3, B, C, 4, D, 5. So our correct answer is C, 4 tablets. So let's take a look at our math here. What's going on? So we have to remember that 1 gram equals 1,000 milligrams. Okay. Each tablet contains 250 milligrams. So the number of tablets needed is 1,000 divided by 250 equals 4 tablets. Okay, you see how that is? So someone's asking about A. So 2 tablets is A. 2 tablets would be 500 milligrams, and we actually want 1 gram. So that's why A isn't the correct answer. So sometimes it might just be useful to just kind of add it up in your head. 1 tablet's 250, 2 tablets is 500, 3 tablets is 750. Oh, 4 tablets, there's my 1,000. Okay. So there's, 2 steps of math going on here. There's that conversion of gram to milligrams, and then figuring out, well, how many milligrams do I need per tablet? So there's a lot of complex math going on here as well. Certainly a question that would be worth really kind of thinking about, because I could see you getting a question like this on the TEAS, either in scientific reasoning or even in math. Okay, now, if you recall, if you were participating in part 1, I merged S4.2 and S4.4 because S4.2 is really interpreting scientific information and 4.4 is evaluating it. And so there's such overlap, it just really made sense to pull them together. So we will have more questions in this section, and they will kind of relate to both interpretation and evaluation. So here we go. Which of the following are characteristics of empirical evidence? Select all that apply. A, based on direct observation or experimentation. B, primarily subjective and influenced by personal opinions. C, measurable and repeatable. D, derived from logical reasoning alone. E, collected using systematic methods. Get those answers out there. So A, C, and E are our correct answers. So empirical evidence based on direct observation or experimentation, measurable and repeatable, collected using systematic methods. So let's take a look at B and D. So what's wrong with B? Why isn't that part of empirical evidence? Because we actually try to avoid subjectivity when it comes to doing scientific experiments. We want to be objective. We don't want to be subjective. And we also try not to have our personal opinions influence our scientific experiments. So this is kind of the complete opposite of empirical evidence. Moving down to D, I would say if the word alone wasn't on there, that would be a good answer, derived from logical reasoning, because you really have to reason logically to even do a scientific experiment. But when you add that word alone in there, that's where our issue is. So even our rationale tells us here, logical reasoning alone does not qualify as empirical evidence because it doesn't involve direct observation or experimentation. But it is very important in forming hypotheses. And it's also important in drawing your conclusions from your data. So just that word alone kind of messed it up. Okay, but A, C, and E were our correct answers. Direct observation and experimentation, measurable and repeatable, and very systematic. We want to do things in a very orderly manner so that when we look at our data, we know that it's based on the one variable we changed and not other factors that we didn't do in a systematic way. All right, let's take a look at our next one. Which of the following lists the typical steps of the scientific method in order? A, experiment, hypothesis, observation, analysis, conclusion. B, observation, hypothesis, experiment, analysis, conclusion. C, hypothesis, observation, experiment, conclusion, analysis. D, analysis, experiment, observation, conclusion, hypothesis. Get those answers out there. Okay, our correct answer is B. So B was observation. You observe something and you think, I wonder why that is. You form that hypothesis. Then based on that hypothesis, you form your experiment. You analyze your data to determine whether your hypothesis was accurate or not. And then you form a conclusion. Okay. Now, at this point, I have someone asking me what empirical means in the reference to empirical evidence. That's a great question. And I would say in that context, it means that it is evidence that has been derived systematically. There's a large body of evidence. It's not just one experiment. There have been multiple studies. So in that context, it's not just saying rely on the evidence, and evidence is one teeny tiny study of 4 people. Empirical evidence is systematic, broad evidence of multiple studies would be the best way that I would use it in that context. So great question. Okay, let's move on now to another question. In an experiment, what is the purpose of the independent variable? A, it remains constant throughout the experiment. B, it is the factor that is measured in response to change. C, it is intentionally changed to observe its effect. Or D, it serves as a baseline for comparison. Okay, our correct answer is C. The independent variable is the factor that we're changing. It's what we're deliberately manipulating. So let's take a look at the others. A, it remains constant throughout the experiment. No, A is standing for all of the other factors that we group into the category called constants that we want to keep exactly the same so that we can ensure that the independent variable is the only thing responsible for any changes we see. Let's take a look at what answer B was. B was, it is the factor that is measured in response to changes. That's the dependent variable, okay? That's what's being measured. That's not what's being manipulated. That's what's being measured. So we had in our content the example of like migraine medicine, and we gave migraine medicine to a group of people who suffered migraines. And the thing that was changing was this medication. And then, or the independent variable is the medication. Then their symptoms, whether their symptoms changed or not, was the dependent variable. All right. And then D, it serves as a baseline for comparison. Remember, that's the control group. So if we go back to the migraine example that we had in our content on Tuesday, we had one group receiving migraine medication, the other group receiving the placebo. Remember, that would be the control group. They would be the baseline to compare it to see if symptoms had improved with the use of that medication. All right, let's do another. All right. Oh, I got some questions. Let's see what some of my questions are. Baseline for comparison means what in an experiment? It means that we want to know if in this case, migraine medicine, I'll stick with that example, if it's actually helping. So if we have a group of, let's say, 20 migraine sufferers, and we put 10 in a group that's actually getting the medication and 10 in a group that's not getting the medication, and we measure their symptoms, we have something to compare it to. So that's what they mean by a baseline. It's something to compare it to. Maybe the group that got the medication still gets migraines, but their pain level is only at a 2 versus 10. But the other group that's not getting the medication still has their pain level at a 10. Well, there's our baseline. We're comparing a 2 and a 10. Okay. Hopefully that helps. All right, let's see what our next one is. Which of the following best describes the purpose of a control group in an experiment? We were just talking about that. A, to receive the treatment being tested. B, to serve as a baseline for comparison. C, to ensure that all variables remain constant, or D, to intentionally manipulate the results. So our correct answer is B, that control group is the baseline. It's what we're comparing it to, okay? So A, the group that's being tested, that is the experimental group. So in our migraine example, the group that actually got the medication is the experimental group. The group that got the placebo is the control group. Okay, let's take a look at C. To ensure all variables remain constant, those are the constants. So sticking with our migraine example, we would want all of our participants to do everything in their normal life the same. We would want them to keep their diet like they normally eat, their sleep patterns like they normally eat, their work schedule like they normally perform, their social life like they normally perform. We would want to keep all of that the same so that we could be sure that the medication's impact was the only thing influencing their migraine symptoms. if they decided on the same day that they started taking the migraine medicine that they were going to start walking every day, or they were going to start doing a new cardiovascular exercise, and they were feeling great. Well, are they feeling great because they're not having migraine symptoms as much, or is the endorphins from the exercise decreasing their migraines? And so that would be confounding. That would kind of skew the results, make the results, we wouldn't know. Is it the medication or is it the exercise? And so we have to keep all those constants the same. All right, D, what was D? To intentionally manipulate the results. No, the control group is not used to manipulate any results, and you don't want to manipulate results. You want results to happen naturally so that you can report your findings from a very objective place. Okay, what is the function of a placebo in medical trials? A, to provide an alternate form of treatment. B, to ensure every participant receives active medication. C, to serve as an inactive substance for comparison with actual treatment. Or D, to measure how different independent variables interact. And the correct answer is C. So the placebo is going to be a harmless, generally it's a sugar pill. It's not an active substance, and it's given to the control group to assess the true effect of the actual treatment. Okay, let's look at our other answers. A, to provide an alternate form of treatment. No, because once again, we wouldn't know then the true impact. We wouldn't even have a baseline to compare that medication to. All right, then let's see what our next one was, B. So B, to ensure every participant receives active medication. Again, if they all get the active medication, we don't have a baseline to compare it to. We don't have that comparison. And then D, to measure how different independent variables interact. No, a placebo is simply used to study how the effects of the treatment is impacting the participants. All right, we're moving right along here. Which of the following are important factors when analyzing the design of an experiment? Select all that apply. A, ensuring a large sample size and repeated trials for reliability. B, keeping all conditions except the independent variable constant. C, allowing participants to choose whether they receive the treatment or placebo. D, maintaining controlled variables across groups. E, changing multiple variables at once to see to see various effects. Sorry about that. All right, let's see what all of our choices were. So our correct answers were A, B, and D. So let's take a look. A, ensuring a large sample size and repeated trials for reliability. So yeah, those are two key factors of ensuring that it is a valid outcome to an experiment. So if we stick with our migraine medication example, we want to have lots of different migraine sufferers taking this medication and testing the results. And we want to have multiple experiments to see if we get the same results each time. Okay. All right, what were some of our others? B was also correct. Keeping all conditions except the independent variable constant. So in our migraine example, like I said, the only thing that would be the thing that we changed was giving the migraine medication. That would be the independent variable. And then we would want everything else same normal, same sleep pattern, same exercise regimen, same diet, same everything, so that we could rely on those results being in reference to the medication and nothing else. D was our other correct answer. Maintaining controlled variables across all groups. Yes, we want to make sure that everything is controlled across all groups. Okay, so that we're not having one group start sleeping 10 hours a night and another group start sleeping 4 hours a night. Now that's going to skew our results. So we have to keep all those things the same. All right, so let's look at our incorrect answers. That makes C one of our first incorrect answers, allowing participants to choose whether they receive the treatment or placebo. We can't do that because the whole point of a placebo is to not have a psychological influence or expectation influence. And what we mean by that is, I talked about this during our content day. If you've ever went into work one day and your good friend, your best friend at work says, "Wow, you're not looking very good today. You feel all right?" And by like 2 or 3 hours later, you're like, "Yeah, wow, I really don't feel good." It was the power of suggestion that influenced you. And so if we allow the participants to choose whether they're getting the placebo or the treatment, they're going to be skewed by whether they're getting treatment or placebo. And so we want to make sure that that's not the case. All right, and then E, changing multiple variables at once to see various effects. That's just like I've been talking about. If we change more than one variable, we don't know if the outcome is dependent on the medication or the change in diet or the change in exercise or the change in our sleep patterns. And so we have to only change one variable at a time. All right, what is the primary purpose of a double-blind study? To ensure neither the participants nor the researchers know who is receiving the treatment or placebo, to allow researchers to decide which participants receive the treatment, to test multiple variables at the same time of day for efficiency, to guarantee that each participant receives, or every participant receives, some form of treatment. Okay, I'm going to leave you hanging for a minute. I've got a question that goes back to what we were talking about in regard to independent variable, dependent variable constants. So my question that was presented was, should our dependent variable be constant? No, our dependent variable is essentially the outcome of our experiment. So if we stick with our migraine medication example, the outcome, whether the patients are experiencing less symptoms, that's our dependent variable. So that should definitely change if our hypothesis is correct. So if our hypothesis is this new migraine medication is going to decrease symptoms, then our dependent variable should show a decreased symptom. If our hypothesis is that and it doesn't show a decreased symptoms, well, now we have to refute it. Now we have to say our hypothesis was not accurate. That medication did not work. So great questions. You guys are really engaged today. I love that. All right, so back to the double-blind study. Our correct answer, are you ready, is A. So a double-blind, and you can see it in the words, is to make sure that neither the participants or the researchers know who's getting the treatment or placebo. That ensures that even researchers can't be biased. Even researchers can't have expectation because they know which group is getting the treatment or the placebo. Okay. And then for B, again, we don't want to hand select who's receiving the treatment. That would skew results. Again, C, if we're testing multiple variables, we don't know what the outcome depended on. And then D, to guarantee every participant receives some form of treatment. No, when participants agree to a situation like that, they're fully aware that they might get a placebo. And that's just them being altruistic and choosing to participate, or maybe they're getting paid to participate. That happens sometimes too. All right. Which of the following guidelines for recognizing, which of the following are guidelines for recognizing bias in an experimental design? Select all that apply. Ensuring participants are randomly assigned to avoid skewed comparisons. B, using blinding, either single or double, to reduce bias from expectations. C, allowing researchers to selectively assign participants to groups. D, being aware of potential conflicts of interests or funding sources that could influence results. Or E, choosing participants based on expected outcomes to confirm hypotheses. Okay, so our correct answers are A, B, and D. Let's break that down. A, so ensuring participants are randomly assigned will help avoid skewing comparisons. Absolutely. Like I was saying in our previous question, we don't want to hand select people. That would skew our results. Okay. B was another correct one. Using blinding, either single or double, to reduce bias from expectations. Absolutely. A strong study would be a double-blind study where neither researchers or participants knew, and we could remove or reduce bias from that. Okay? All right, our other correct answer was D. Let's take a look at D. Being aware of potential conflicts of interest or funding sources that could influence results. Yes, I talked about this on Tuesday. If the company that is trying out their new migraine medication is paying for the experiment, if they're funding the lab, if they're funding the equipment and all of that, well, there's probably some conflict of interest there. There's probably going to be some bias that comes in there. So we want to be aware of who paid for that. All right, let's see what our next incorrect answer would be C and E. So C says allowing researchers to selectively assign participants to groups. No. Why? Because that would skew our results. E, choosing participants based on expected outcomes to confirm hypotheses. No, we can't do that. We can't just mold the experiment to go the way we want it to go. That would not be an ethical experiment. That would not actually really teach us anything. We are looking for true objective data. All right. A researcher is studying the effect of a new allergy medication on symptom severity. One group of patients takes the new medication, while another group does not. After 2 weeks, the researcher records the severity of allergy symptoms in each participant. Which of the following correctly identifies the independent and dependent variables in this experiment? A, independent variable, allergy symptom severity. B, I'm sorry, dependent variable would be part of A, whether or not the patient takes the medication. B, independent variable, administration of the medication. Dependent variable, allergy symptom severity. C, independent variable, time of day the medication was taken. Dependent variable, type of allergy medication used. D, independent variable, pre-existing allergy conditions, or dependent variable, dosage of medication given. Our correct answer is B. So let's take a look. Independent variable, the medication, right? The administration of giving the groups their medication. Dependent variable, how their allergy symptoms are in response to that medication. Okay. So A, what's wrong with that? Well, the independent variable is what you're purposefully manipulating. Are we purposefully manipulating the symptoms of their allergies? No, we're trying to see if the medication will do that, but we're not doing that. Okay. C was talking about time of day and type of allergy medication. Those were not relevant to the question. The question was really assessing, does this new allergy medication decrease severity? Okay. Same thing here. Pre-existing allergy conditions, that's not something we can manipulate. And remember, we manipulate the independent variable. Dependent variable, dosage of medication given. No, that dependent variable is, will their symptoms change? Okay. So that's what we want to keep in mind. Independent variable is what we're manipulating, is what we're changing. Dependent variable is essentially the outcome. What happens when we make this change? There's a cause and effect there. If we make this change, do we have this effect? All right, we are on to our last category, predicting relationships. This would align with S4.3. Which of the following lists the levels of biological organization in the correct order from smallest to largest? And I'm just going to leave the screen and let you look at it for a minute before I set the timer. I think if I read through it, it's going to be more confusing. So I'm going to give you like 10 seconds and then I'll set the timer. Okay, our correct answer is C. Okay, and I pulled this image from our content from Tuesday. So from smallest to largest, we have atoms that make up molecules, molecules that make up the organelles which fit inside the cells. Cells work together to form tissues. Tissues work together to form organs. Organs work together to form the organ system, and then all the organ systems work together to form the organism. Okay. So what was wrong with the other answers? Well, atom is smaller than molecule. So that right there made A incorrect. Then let's see what our next one was. C was our correct answer. So B, atom, molecule, cell, organelles are inside cells. One of the ways that you can kind of remember that is we know organs are inside our bodies. So organelles are like little tiny organs inside the cell. Okay. And then D, right out of the gate, we see also that atom is smaller than molecule, organelle is smaller than cell, and so that made that one incorrect as well. All right. I think someone asked if A and C were the same. No, because we can see here that molecules and atoms are switched, and that right there is what made those not the same. Okay, let's see what our next one is. How does the sequence of events in a biological process demonstrate a cause and effect relationship? A, each event happens randomly without influencing the next step. B, earlier events directly influence or determine the outcome of later events. C, later events influence earlier events in the sequence. Or D, all events happen simultaneously with no connection between them. Get those answers out there. Our correct answer is B. Okay, so in a cause and effect relationship, those earlier events are going to lead to outcomes. So the sequence of events shows how one step influences the next. So this talks about what we just looked at in our previous question. So in biological organizations, cells form tissues, which build organs, which then build the organ systems, and so on. So let's take a look at the other ones. Each event happens randomly without influencing the next step, not if we're looking at a sequence of events in a biological process. Each event is going to influence what comes after that. C was just kind of silly. How will later events influence earlier events? That's kind of backwards. And then if it's a sequence of events, then they're not going to just happen simultaneously without connection, not if we've identified it as a sequence of events. All right. Which of the following correctly describes the sequence of events in a feedback loop? And again, I think when these have the arrows in it, it's probably more confusing if I read it. So I'm going to just be quiet, let you read through it, and I'm going to give us about 10 extra seconds. Okay. Our correct answer was B. And I pulled this visual from our content. So we've got our initial stimulus that's going to cause some sort of change, or we got a variable there. Receptors in our body are going to detect that change, send that information to a control center. Typically, it's the brain, a spot in the brain. That's going to send out some sort of effect. It's going to send out and say, we've got to alter that variable. Okay. And that alteration will depend on whether it's a negative or a positive feedback loop, which we're going to get into. So I don't want to give any of those answers away. So if we look at our answers, we can see right out of the gate that it wasn't logical because the control center is evaluating a change before we even detect a change. So that was kind of a rule that one out, because how are we evaluating a change that we haven't detected yet? C, the effectors are altering the variable. We haven't even detected that there was a change in the variable yet. So again, that makes that incorrect. And then D, receptor detects change. Okay, effectors alter the change. Control center evaluates the change, and then the initial stimulus, well, the initial stimulus is what kicks it all off in the first place. And so that's why that would be incorrect as well. We've got to start with that initial stimulus. Now one of our next questions has to do with like what type of feedback loop and what that stimulus would be. So we're gonna kind of put a pin in any question on that just for a moment. All right, so here we go. Which of the following statements correctly describe positive and negative feedback loops? Select all that apply. And this is why I didn't want to go over each of them, because I didn't want to give your answers away. So let's take a look at our choices. Negative feedback loops work to restore stability by reversing a change in a variable. B, positive feedback loops enhance or amplify a change until a specific outcome is reached. C, negative feedback loops push the body farther away from homeostasis. D, positive feedback loops are more common in maintaining daily physiological balance. E, negative feedback loops help regulate body temperature, blood sugar levels, and blood pressure. Get those answers out there. This is our last one, by the way, so finish strong. All right, so let's look at what all of our correct answers were. A, B, and E. So let's break it down. Negative feedback loops work to restore stability by reversing a change in the variable. So let's go back to our visual here. So in this case, when we come around this loop here, this effector is going to bring this variable back up to a balanced place. Okay, in a negative feedback mechanism, whatever is going on here is going to bring this back to a balanced place. Let's say, for example, it was temperature. It's really cold outside, so our body temperature drops. Receptors in our skin detect that. Signals get sent to our brain. Our brain sends out signals to shiver, to constrict our blood vessels, to help get our body temperature back to 98.6 degrees. Okay? All right, what was our next correct one? B. Positive feedback loops enhance or amplify a change until a specific outcome is reached. So in a positive feedback loop, we have all of this is the same except the effectors. In this case, it's going to enhance what's going on. For example, we had in our content childbirth. If the baby is pushing on the cervix, stretch receptors are going to detect that, send that signal to the brain, which is going to release oxytocin. Oxytocin causes uterine contractions, which puts more pressure on the cervix and enhances that pressure. And that's going to keep going until the baby is born. That's a positive feedback loop. So where the difference is right here, the effectors and what they're doing to that variable. All right, our next correct answer was E. Negative feedback loops help regulate body temperature, blood sugar levels, and blood pressure. Yes. So negative feedback loops are the most common loops going on in our body because our body wants to be in homeostasis. You know, everything in our body should be in a homeostatic place. And so we're constantly trying to keep everything balanced. Okay. And so negative feedback loops are working to bring this variable back to its set point. Blood sugar levels drop, it's going to bring it back to its set point. If we're too thirsty, we're dehydrated, it's going to try to, send out thirst signals for us to drink water. So everything is about balance in that regard. All right, so we went over the correct answer. So let's look at C, our incorrect answer. What's wrong with this? Negative feedback loops push the body farther away from homeostasis. No, positive feedback loops do to a set point. Okay, when the baby's born, then that loop is done. Another situation would be if you got a cut. The stuff that's at the cut is going to send out signals to keep coming to block that cut to help you clot. And then once you're clotted, once you're no longer bleeding, then that feedback loop would be done. Okay, and then what was our other incorrect answer? D. Positive feedback loops are more common in maintaining daily physiological balance. No. Positive feedback loops are for a particular reason, childbirth, bleeding, things like that. Negative feedback loops are our everyday body loops that are going on constantly. Okay? All right. Oh, I lied. I'm so sorry. I thought we were on our last one. Normally, I have multiple screens up to where I can see what's coming, but with me streaming to Instagram, that's got my other screen. And so I was going off of memory. So I apologize. I believe this is our last one, though. All right. Which of the following scenarios is an example of a negative feedback mechanism? A, during childbirth, contractions increase intensity until the baby is delivered. B, when body temperature rises, sweat glands activate to cool the body down. C, a blood clot forms rapidly at the site of an injury to stop excessive bleeding. Or D, the release of oxytocin during breastfeeding continues as the baby nurses. Get those answers in there. So sorry I lied. Didn't mean to. Our correct answer is B. Okay, the body temperature rises, sweat glands activate to cool down the body, bringing us back to that 98.6 degrees. So again, going back to balance. A, C, and D, these are all positive feedback mechanisms. Childbirth, blood clotting, I talked about those. Release of oxytocin during breastfeeding, those are all positive feedback mechanisms. So I did want to note, someone commented in the feed that select all that apply is very stressful. Agreed. And that's a lot of feedback when I've tutored. I've heard people say that that's really kind of what stress them out during the TEAS. So, we'd like to give you a similar environment so that you're comfortable with that when you actually take the TEAS. So, but I agree with you. It's much more stressful. All right, so I always like to bring us back to these just in case you missed the QR codes at the beginning. Remember, you've got free live webinars. You can rewatch even today's webinar. That question bank now is over 2,000 questions. I'm so excited. On-demand videos, I showed you guys those at the beginning. They're the bomb. You get cheat sheets, you get the slides. Tutoring, I would love to tutor any of you. I had a tutor session yesterday and it was just fantastic. You guys are so eager to learn so that you can be successful, and I love that. So reach out and schedule a tutoring session. Remember our SurePass and SurePass Pro, if you package things, you get a better price. And then catch us on all the socials. And please, please, please give me some feedback. I want to know how I did today and how you're liking our new format of content and then questioning. And just as an FYI, my next webinar will be biology in April, on April 7th at 3 o'clock. And we're actually breaking the biology in two. So that will be like macromolecules and cells. And then I'll have another one on the 14th that will be genetics and like the laws of, what do I want to say? The Mendel's laws. That's what I'm going for. And they will be this setup where we have content on one day and then we have the practice test on the other day. So I certainly hope that you guys will come see me again, and best of luck as you're studying. Please send me some feedback and have a fantastic day. Take care.